Now that we're done talking about amino acids individually, we're going to take a more holistic approach to proteins. So we're going to start by talking about the 4 distinct levels of protein structure and how each of those contributes to the proper folding of a protein. Then we're going to talk about what happens if that doesn't happen and we actually have a misfolded protein, and we're going to end off with some examples of protein function. So as this picture shows, the 4 stages of protein structure really dictate the function of a given protein. And we can see here that proteins are super multifunctional. They're involved in regulation, structure, movement, catalysis, transport, and signaling. Each of the 4 levels has a corresponding particular type of bond, so we're going to dive into each of those now. So to start, the primary structure of a protein is essentially just the linear sequence of the amino acids. So it's basically just the polypeptide chain. And as we learned in the last lecture, the bond involved in polypeptides is that peptide bond. So you see here's the N terminus, and this is our long polypeptide chain ending in the C terminus. And then here we see that each amino acid is connected to another one via a peptide bond. If you recall from last lecture, we talked about the 3 stages that a amino acid can go through based on the relationship between pH and the pKa of each of the groups on that amino acid. So now we're going to apply the same principles so that we can find the net charge of a polypeptide chain. So as an example, we're going to find the net charge of a polypeptide that is defined by the single letters KILT, and we're going to do that at a pH of 4. So the first step is to label your chain with all of the pKa values. So I just wrote out, I know K is the first amino acid, so that's going to have the amino group attached to it, and T is the last one, so that's going to have the carboxylic group attached to it. And then I just looked at this chart that we talked about last lecture, and I found each of the corresponding pKa groups where they're relevant. So K is lysine, so you can go here and find lysine down here. The amino group, which we know should be around 9, for lysine is 8.95, so I marked that under the N. And then lysine also has a R group with a pKa value because it's basic, so that's a 10.53, that's under the K. I stands for isoleucine, L is for leucine, and T is for threonine, and as we know, these are neither acidic nor basic, so none of them actually have a third pKa value, so that's why they're all empty under it. However, T, because it's the last amino acid and has that carboxylic acid, or carboxyl group attached to it, we do need to find its pKa value. So I look for T, threonine, and that carboxyl group should be around 2, which it is, 2.09. So I've put that right underneath the C. Next, we're going to pretend that the pH of our solution is super, super low. So pretend it's 1, and we're going to protonate and deprotonate accordingly. So if the pH was 1, we know that that would be less than 8.95. Therefore, the group would be protonated. So we're going to protonate that N, and I know when the amino group is protonated, it becomes NH3 positive. Then we're going to do the same thing looking at the K, so it has a pKa of 10.53. That, again, the pH of the solution, which is 1, is less than 10.53, so it gets protonated. K stands for lysine, which is basic, so we know anything basic and gets protonated, similar to NH3, becomes positive. Then we can skip I, L, and T side groups because none of them have relevant pKa values, but the carboxyl group at the end of T, again, 2 is greater than a pH of 1, so it gets protonated. Carboxyl groups, however, when they're protonated, they don't get positive, they just become neutral. Now that we're in the fully protonated stage, we're going to actually adjust our pH to the one mentioned in the question, which is 4, and we're going to protonate and deprotonate accordingly. So I've just rewrote what we had at the end of step 2 in green, and now as we go through each of the side groups, we're going to update it. So now our pH is 4. 4 is still less than 8.95, So NH3 remains protonated. We leave that one alone. The next group is the 10.53. Again, the pH is smaller, so it stays protonated. And then we skip I, L, and T, and we go to our carboxyl group. Now the pH of 4 is actually greater than the pKa of 2 of that group, and so it's going to be deprotonated. And so that's what happened here. The H came off, and now it's negative. And then your last step is actually to just add up all of your charges and find the net charge. So at a pH of 4, we know that these two will be protonated, and this one will be deprotonated. So we have positive, positive, negative. That leads to a net charge of +1. So that's how you approach questions like this. Another type of question that can be asked about the primary structure of proteins, which is just that polypeptide chain, is what is the isoelectric point? So we know that the isoelectric point is basically when the net charge on the polypeptide is 0. So that's what we're going to look out for. So the first step, just like the last question, we're just going to label our chain with our corresponding pKa values. So in this question, they're asking about the polypeptide DREN, and so I've listed it out again with the amino group and the carboxyl group, and I've listed the corresponding pKa values based off of this chart. Notice D, R, and E all have pKa's, and that's because they're part of DERK, right? Our acidic and basic amino acids. So what I like to do when I'm going to label these, just to make it a little bit faster, I call it like a DERK scan. Basically, I just look at each and I see like, are you either of those 5 letters? And if they are, they have a pKa. If not, for example, N, it's just blank. The next step for this type of question is the same as the last. We're going to pretend our pH is super, super low at 1, and we're going to protonate things accordingly. We're then going to get the net charge at that low pH. So starting with the first one, the N, the pH of 1 would be less than this, so this would get protonated. Same for D, 1 is less than 3.65. Note though, D is a acid. So just like the carboxyl group, when it is protonated, it has an H, but it's neutral. Only the amino and basic amino acids become positive. Then we move on to the R. Again, pH 1 is less, so it gets protonated. R is basic, right, arginine, and so that has a positive charge. E then is acidic. It will also get protonated because 1 is less than 4. However, because it's an acid, we just add a neutral H. The N side group we can ignore, and then the carboxyl group will also be protonated, and that will just have that H. And so our net charge at pH of 1 is going to be positive 2. We have these 2 positives, and we have no negatives to counteract that. Unlike the last question that told us exactly what pH we need to go to, we're actually going to slowly increase our pH, and we're going to make changes accordingly as we go. So right here, I've just kind of copied down the final result of step 2, and we knew that that was at a pH of 1. We had a charge of positive 2. Now let's say we increase the pH until we hit our first pKa value, which is going to be at 2.02. When we go beyond 2.02, the pH will then be greater than this group's pKa, and so the group will be deprotonated. So that H will then come off, and that'll leave us with a negative charge here. So now I've just noted we started at a +2 when we were at a pH of around 1, but once we hit 2.02, we now have a net charge of positive 1 because this one balances out one of them. Now we're just going to continue to repeat the process. So we're going to continue to increase the pH until our next pKa, which is 3.65. Once we are a pH greater than this, we will then deprotonate that group. Again, D stands for aspartic acid, and so it will be deprotonated and become negative. That leaves us with a net charge of 0. Now we're going to do it again. So we increase until the next pKa, which is 4.25, and that has to do with the side group of E, which is glutamic acid. when that one gets deprotonated, the net charge becomes -1. Then we do it again, and you just keep on doing this until you are all the way at the highest pKa. And so we'll go up to 9.6, deprotonate. Amino groups are normally basic, so when they get deprotonated, they're neutral. Net charge is -2. And then we do it again to 12.48. That will also become neutral, and so our net charge becomes -3. But remember, we're looking for the isoelectric point, which is when our net charge is 0. So even though we went all the way for this example, you actually can stop once you hit -1, and that's because we just want the 2 relevant pKa's that the average would lead me perfectly to a net charge of 0. So I noticed that the 2 corresponding pKa's are 3.65 and 4.25. You average the 2, and then you know your pi, your isoelectric point, is 3.95. So that's how you approach questions like this. So the next level of protein structure is the secondary level. This is really just local folding of polypeptide chains that's happening because the backbone atoms are actually going to be forming hydrogen bonds. And so we have 2 types. We have the alpha helix, which kind of is similar to how DNA looks. And what you want to know about this is that the side chains are actually going to radiate outwards, right? Secondary structure is all about the backbone. So the backbone is what actually makes the helical shape, and then the side chains will just kind of pop out to the left and right along the way. Also, because the angles of the loops are all the same, these are going to tend to show repeating patterns of side chain polarity, and that's going to happen about every 3 to 4 residues. On the other hand, we have the beta-pleated sheets. These are more of a flat, kind of in one plane version. So our NCC NCC backbones, that's going to make the plane of, it's kind of like rows, and that's going to make the plane of, imagine like a paper. So instead, the side chains are actually going to radiate off the paper. So it'll go either if you're holding the paper flat on your desk, it would go straight up or straight down. Now, within a beta-pleated sheet, we have 2 different types. There's the anti-parallel and parallel, and that has to do with the direction that the rows within the sheet are moving. So in anti-parallel, they're going in opposite directions. So you can see this first one would be from the N to the C, and then the next one the opposite way. N would start on the left and go to the C on the right. In parallel, they're both going the same direction, so both of these are going from N to C. Another difference you want to note between the 2, in anti-parallel, you can see here I've marked it, but one amino acid is going to touch one other amino acid in the row next to them. For parallel, one amino acid will actually bind to 2 separate amino acids in the row next to them. Also, in comparison to the pattern of the alpha helix, for the beta-pleated sheets, they actually show patterns of polarity every other residue. The next thing about secondary structure is that the peptide bond is actually planar, and so what's going to happen is polypeptides can rotate in some places and not in others, and that's due to the steric hindrance that comes with the structure. So what we have are these 2 bonds. One is known as the phi and one is known as the psi. And these are the 2 bonds right next to the alpha carbon. So one's going to be between the N and the first C, and the other one would be between those 2 C's for any given amino acid. And these are actually going to restrict how much the polypeptide can rotate, and that's because it's sometimes the 2 would occupy the same space, which we don't want. And so then we have the Ramachandran plot, which actually is a visual representation of how much a polypeptide can rotate. And so it shows the sterically allowed phi's and psi angles, and you're going to see that, as you can see here, for glycine, so basically all these dots, those are the more the dots, the more options you have to rotate. So something like glycine, which has a really small side chain, is going to have way more flexibility than something like proline, which one has a big R group and also it's more restricted. So just keep that in mind. So some proteins really only need to go as far as the secondary structure, and those are what we call fibrous proteins. So they're going to be elongated molecules that have really well-defined secondary structures. They tend to have a structural function, and that's why they don't really need anything more than this rigid type of shape. Some examples are keratin, which you find in like your hair, your fingernails, things like that. Then there's collagen, which is a connective tissue protein, and that plays a role in our like bones. And then there's also something known as fibroin, which is actually in silk cocoons. So you can see here, keratin and collagen both had alpha helical structures, but then the fibroin has the beta pleated sheets, and so you see these planes of rows happening. The opposite of fibrous proteins would be globular proteins. These have more of a sphere-like shape, and so they're going to require a tertiary structure, sometimes even a quaternary structure, to really fold it more into the proper shape. These are generally soluble in water, and we'll talk about why, and they're also super diverse, and that's where the majority of our proteins are going to come from. So these are some examples and how they're all very different. The tertiary structure is more of a distant folding rather than local folding, and it's all about side chains. So we're going to have side chains maybe that are further down the polypeptide chain that are going to interact with one another and cause the protein to fold in a certain way. There's not exactly one bond like the primary and secondary structures that define tertiary structure, but there's actually a lot of different options. So we can have hydrophobic interactions, and that could be come in the way of van der Waals bonds, and that would occur between nonpolar side chains. Or we can have hydrophilic interactions, so we'd have hydrogen bonds between polar side chains. Or you could have electrostatic interactions, and that would be ionic bonds between acids and basic amino acids. So here's an example. So here's our polypeptide chain. Here we have an amino acid here, and then one a lot further down the line over here. They're making a hydrogen bond. We also have hydrophobic interactions, ionic bonds, and then there's actually also disulfide bridges, which are an example of the covalent bonds that can cause the protein to fold. So the tertiary structure is all about interactions between side chains, and that's going to be of amino acids that are actually located a little bit further down the line in the polypeptide chain. And unlike the primary and secondary levels, tertiary does not have a single bond that defines it, but there's actually a lot of different options. And so we're going to break it down into non-covalent and covalent. Many of them, most of them, are non-covalent. So we can have hydrophobic interactions. That would be in the form of a van der Waals bond between non-polar side chains. So here we have some hydrophobic interactions, and note like one amino acid would be here, the other one would be quite further down the line, all the way over here. Then we have hydrophilic interactions that could be coming in the shape of hydrogen bonds between polar side groups. That's happening here. And then there's also electrostatic interactions, which would be in the form of ionic bonds between acidic and basic amino acids, which is shown down here. Then we also have a specific type of a covalent bond that's very prevalent in tertiary structure, and that's something called a disulfide bridge. So let's walk. So what a disulfide bond basically is just two cysteine amino acids. Here I have one, here I have another, and their sulfhydryl groups are actually going to form a bond, and that's going to occur via oxidation because we're losing the hydrogens. And then your final product is going to be a cysteine directly attached to another one, and this new overall structure is actually called cysteine. And so you can see that in action on a polypeptide here. We have one cysteine up here and then one a lot further down the line over here. They're going to attach to each other, forming that disulfide bond. You could also go backwards and you could reduce it by breaking that bond. An interesting phenomenon that we've kind of talked about before, but now in direct relation to proteins, is the hydrophobic effect. So basically what this is the the polypeptide will actually fold in a specific way that's going to make sure that all those non-polar side chains, they're going to be on the inside of the, like, let's call it a blob, right? And then all the polar side chains, they're going to be on the outside of the protein, and that's so that they can interact maybe with like water or something that's a lot more polar, the solution around them. And those non-polar ones, they can form bonds with each other, and now they're safe from any polar regions. And this hydrophobic effect is actually super important in terms of the thermodynamics of protein folding. So just overall, in terms of proteins, favorable things that we have going for us in terms of thermo. So we have those intramolecular enthalpic interactions, and that's basically all those, the bonds that we talked about for tertiary. So those nonpolar, polar, hydrophilic, hydrophobic, all of those interactions between side chains. That's good. We want that. What's unfavorable for us is the fact that going from secondary to tertiary, we're actually ordering our protein a little bit more. And so if you remember from our thermo lecture, that would lead to a lower entropy, right? We want things to be more disordered. However, we're doing the exact opposite here. So that's unfavorable for us. What happens though is the hydrophobic effect is actually the solution to that whole thing. And that's because when we have these side chains all out in the open, water is able to orderly form around each side chain. But when we glob all of the nonpolar regions together in the center, now the water is just around one giant thing. So it's not really ordering around 4 in this picture, it's ordering more around 1, and so it's a higher entropy. The last level of protein structure is the quaternary level, and this is essentially the same as the tertiary, except for the fact that it's no longer on one polypeptide, but it's going to be interactions that are occurring between separate polypeptide chains. The bonds, however, are the same as the tertiary because there are so many of them. What we call a subunit is if we have multiple polypeptides coming together to form an overall quaternary structure, each of the individual ones are considered a separate subunit. So this is an example we see in hemoglobin, and we'll talk more about the details of this specific protein in a little bit, but I just want to make it clear that these quaternary interactions between the subunits are actually instrumental in protein function. So because of the way that they interact with each other, we get something that we call cooperative binding of oxygen, which is essentially once one oxygen binds to a heme group, the next one is more likely to bind it. And we'll talk about that more in the next lecture actually as well. So this is the picture of complete protein structure, right? As a review, we have our primary one, which is just the polypeptide sequence. They're all attached via peptide bonds. Then we have local folding, which is the secondary structure, and that's going to be characterized by hydrogen bonds. Here in this example, they use an alpha helix, but remember it could also be a beta pleated sheet. Then we fold this a little bit further and we get the tertiary structure, and that's going to be due to interactions between our side chains. Plenty of bonds that can do that. And quaternary structure is going to be when we take a bunch of these folded tertiary polypeptides and attach them together with the same types of interactions that you can use in tertiary. Sometimes a protein won't properly fold or it'll be disrupted, and that can happen via denaturation. So this is the disruption of a protein shape, but you're not actually breaking the peptide bonds. So technically your primary structure is still intact, it's just the secondary or tertiary and maybe even the quaternary structure that are interrupted. And so here we have a picture, here's our properly folded protein, but it's going to get denatured. So you still see that primary one, just not the secondary and tertiary. Or if this was the subunit quaternary. And so because it's unfolded now, it's going to lose its function. Factors that can cause denaturation have to do with if we have an extreme in temperature or pH. So most proteins only operate within a certain range of those two factors. Also, there can be changes in salt concentrations or the tonicity. That can also lead to denaturation, as well as things like urea, which actively break hydrogen bonds. So that would affect the secondary structure, or even tertiary and quaternary as well. Obviously, we want things to be folded properly, and a big reason is misfolding can actually cause certain diseases. So we talked about how proteins get translated right at ribosomes. When they get translated from the primary to secondary structure, that actually happens really, really quickly. It's almost a matter of seconds. But then going from the secondary structure to the tertiary is a lot slower. It could take up to a couple minutes. And so in that process, when it's going from secondary to tertiary, sometimes we'll end up with misfolded proteins, meaning that their tertiary structure is not properly intact. And so what happens here is the proteins will have hydrophobic regions that are going to be exposed, and then the more of those, they're going to actually cause, they're going to clump together and form aggregates, right? Nonpolar to nonpolar, they want to clump together. And that is actually really, really bad, because those aggregates can cause things like amyloid plaques or neurofibril tangles, and those are ways that we get Alzheimer's disease. So this picture is showing 3 different diseases. We have Alzheimer's, Parkinson's, and Huntington's. The top row is going to be what the protein should look like, but then the bottom row shows what happens if they get misfolded and form those aggregates. Those are what's going to lead to the diseases. And the picture below is just showing you additional versions of proteins that can be misfolded. So I know for Alzheimer's up here it mentioned amyloid and tau, but then there's also mad cow disease and another one that have other misfolded proteins. Also note, this is actually a common cause for a lot of neurodegenerative diseases, right? Alzheimer's, Parkinson's, Huntington's, things like that. The solution, however, is to try to minimize misfolding, and that's going to come in the shape of things called chaperones and proteasomes. So a chaperone is essentially just a protein that's going to promote and try to help make sure proteins are folded properly. So an example is the GRO-EL and ES chaperonin, and it's going to act on fully synthesized proteins. What it's going to do is it's going to essentially form kind of like a barrel to isolate the protein and then make sure it has good conditions to refold itself. So down here you'll see we have 2 barrels. The top one is considered the cis ring and the bottom is the trans. So what will first happen is one of the barrels on the GRO-EL, so in this case the top one, the cis one, is going to capture a misfolded protein. Then what's going to happen is the cap, GRO-ES, that's why it's a pair, is going to attach along with ATP, and that's going to cause that ring to actually increase in diameter and make more space in there, allowing the misfolded protein to unfold and then refold itself properly. Once it is nicely refolded into the way it should be, ATP is going to get hydrolyzed, and that's going to weaken the complex in terms of the binding between the cap and the cis barrel. Then a new misfolded protein will attach to the other barrel, or in this case the trans ring, along with ATP. The cis ring will then release its cap along with a DP, the leftover version of that hydrolyzed ATP, and the cap will come off, the ADP and the newly properly folded protein comes off. And then what happens is the whole system actually does a 180 turn. So what was my cis ring that is now empty and what was my trans ring that has the misproperly new folded protein is going to flip, and so now the misfolded, the new one, is going to be at the top, and that's going to be our new cis ring, and the bottom one, the trans one, will be empty, and then we just restart the whole process. So that's kind of how chaperones work. Unlike chaperones that are there to help refold things, proteasomes are actually protein complexes that degrade unneeded and damaged proteins. They're going to do this via proteolysis. So they're going to break a bond, a peptide bond, one at a time, and kind of eat away at the polypeptide chain. And that's kind of, imagine it, an analogy is like Pac-Man eating away things. And so what's going to happen is when we have a misfolded protein, what's going to happen is that the chaperones and the proteasomes are actually going to kind of like fight over it. And so then the longer a protein remains misfolded, meaning that maybe the chaperones haven't gotten around to it and hasn't been refolded properly, the more likely it's going to get caught by a proteasome and then be completely degraded. And this is better than it existing and forming those aggregates that would lead to diseases. So that's enough about protein structure. Now we're going to go into the different functions that they have. So we talked about it before, but proteins are multifunctional. They do plenty of things within our bodies. For us, we're going to focus on three things. We're going to talk about the contractile functions of actin and myosin within our muscles, and then antibodies and how that keeps us safe from foreign toxins, and hemoglobin for the transport of our oxygen. So first off, we have hemoglobin, which is a protein that is in our blood and helps us transport oxygen from our lungs all the way to all of our tissues. Hemoglobin is a protein complex, so it's made up of 4 polypeptide subunits, so it does have quaternary level of structure to it, and each of those subunits actually has a heme group with an oxygen binding site. So here we have 1, 2, 3, 4 polypeptides, 4 heme groups. Each of them is going to attach to oxygen. The thing about hemoglobin is that it follows cooperativity, so that means that once one of those oxygens binds to one of those subunits, the others are going to be more readily able to accept more oxygen. And so yeah, we're gonna bring in the oxygen from the lungs. Once one comes, more comes, and then we will travel to our tissues and release it there. Our next example is muscle contraction. So within our muscles, they're made of repeating units of bundles of muscle fibers, which actually have myofibrils. And each myofibril has repeating units that we call a sarcomere. So here's a picture of a sarcomere, and actually within each sarcomere we have 2 different proteins at action. One is the myosin, that's going to be this thick red filament, and then we have the actin, which is the thinner yellow filament. And actually the alteration between the thick and thin is what leads our muscles to look striated. So within one sarcomere we have the center line, which is called the M line, and then on the edges we have Z lines. And basically the sarcomere will shorten from both sides as the actin filaments slide along the myosin filaments, and that's actually what leads to our next slide, which is the sliding filament model. So within a sarcomere, we're going to contract and shorten that sarcomere distance. The way we do that is the myosin head is actually going to attach to the actin filament, and then it's going to do what is called a working stroke, and it's actually going to pull the actin filament to the left, and so you see here it's attached, and then it pulls it over this way, and then an ATP molecule is going to come in and allow it to kind of let go of that actin filament, and then it's going to break down, allowing it to go back to the ready state to reattach to another site. And then it'll just repeat all over again, and it's going to pull and let go, and then attach to a new site, pull, let go. Last up, we have the immune response. And so we have antibodies within our immune system. They're basically blood proteins that are produced in response to counteracting anything bad. And those bad things are what we call antigens. So they can come in the form of viruses, fungi, bacteria, toxins, chemicals, basically anything bad. Antibodies are going to try to act against it. And so this is the structure of an antibody. It has 4 different polypeptide subunits. 2 of them are what are called light chains, and basically that just means that they're smaller. And then the other 2 are called heavy chains, and they're just a little bit bigger. At the 2 ends of the antibodies, we have binding sites for the antigen. And so these are very, very specific. So even though we have, could have like many antigens, only one will fit to a specific antibody. We also have disulfide bridges, which are just going to hold the whole structure together. And so one antibody is going to bind to 2 antigens, and that's going to stop those antigens from entering and damaging cells. So in a way, antibodies are basically neutralizing antigens. Also, there are millions of antibodies within our body, and they all have slightly different structures. So we have they can recognize a diverse range of antigens and take care of them. And this is just a summary slide of the overall structure of a protein. So these are the main 4 levels and their different characteristics and interactions respectively. Then these are the 2 types of the general shape proteins, right? We have those fibrous ones, which are really just secondary, and then the rest, which are globular. And then we have things that can combat misfolding, right? Because if things misfold, we're going to lead to different diseases. So we have chaperones that are fighting against that. We also have proteasomes, and then it goes into all the different biological functions that proteins have.